History of microbiology. Today we are going to discuss the history of microbiology. Medical microbiology is an important topic in modern medical science. Today we will briefly focus on the golden age of microbiology and highlight the contribution of some key scientists who made groundbreaking discoveries in the field of microbiology. Invisible living creatures produce disease. In the early period, before the invention of microscope or before the golden age of microbiology, people believed that some invisible living things or organism or something is there, some invisible things are there, and they are responsible for causing infection or disease. Varro and Columella in the first century BC postulated that disease was caused by invisible being called Animalia minuta. Whereas in 2nd century BC recorded principles of contagion by invisible creature. The contagion means nothing but the transfer of infection from one person to another, or we can say that spreading of infection is called as contagion. And Roger Bacon in 13th century postulated invisible living creature produce disease, and Franciscus of Verona 1546 pointed out that communicable disease was caused by germs or these invisible organisms. Golden Age of Microbiology. Golden Age of Microbiology, 1815s and early 19s, was a key period when major discoveries sharpened in modern microbiology. Scientists like Louis Pasteur, Robert Koch, and Joseph Lister made significant progress in understanding how microorganisms cause infections or disease. Pasteur showed how microbes are linked to disease and developed vaccines. Koch created a method to identify disease-causing bacteria. Lister introduced antiseptic technique in surgery. This period is also saw the creations of essential tool like agar plate of growing bacteria, and the same agar plate we are still using today. This achievement improved our understanding of infectious disease, leading to antibiotic vaccines and modern medical practices, transforming medicine and public health. Altogether, we can say that in this golden age of microbiology, mainly the scientists like Louis Pasteur, Robert Koch, and Joseph Lister, they have made many contributions to the field of microbiology that has given a baseline for the development of medical microbiology in the later period. Discovery of microscope. Antony van Leeuwenhoek first observed in 1673 and described living microorganism by his microscope. So the microscope was discovered by Antony van Leeuwenhoek in 1673. His hobby was grinding lens and observing diverse material through them. So he used to grind the lens, and through that lens he will observe the microorganisms. He observed in peat scraping, rainwater, and peppercorn infusions. And by his microscope, he used to take this teeth scraping, rainwater, and all, and he will observe those things through his microscope. He first observed Giardia lampiae in his stool in 1681. In 1678, Robert Hooke developed a compound microscope and confirmed Leeuwenhoek finding. And this is the microscope invented by Antony van Leeuwenhoek. And this is the length. These two are the lengths, and this is the specimen keeping area, and these two are the screws. And by these screws, first he will keep the specimen here, and by these screw, he used to adjust the specimen towards this length, and he observed the specimen. He used to observe the specimen through his this length. Okay, and after that, and he recorded his observation like this. and these two are the rod-shaped bacteria, and this is also a long rod-shaped bacteria, and E, this is the cocci or spherical shaped bacteria, and G, this is the spiral shaped bacteria, and this is the spherical shaped bacteria arranged in clusters. And after that, he reported his observation to the Royal Society of London in 1676. But during that time, they are not given much important for this observation. Louis Pasteur. Louis Pasteur is a French chemist known as the father of modern microbiology. And why he is called as a father of modern microbiology? Because of his various contribution in the field of microbiology during the golden age or golden period of microbiology. In 1857, he established that fermentation is caused by microbial activity. That means you can say that fermentation is occurred due to the bacterial or microbial activity, or the fermentation is the process of fermentation is occurring due to the activity of bacteria or microorganisms. And he introduced the technique of sterilization such as steam sterilizer, autoclave, hot air oven, flaming, and pasteurization. And we can say that still in our microbiology laboratory, we are using these sterilization method. And he also established that the importance of using cotton plug to protect culture media from aerial contamination. Aerial contamination means air contamination. That means this cotton plug is used to cover or plug the uh, test tube. That means by applying this cotton plug to the test tube, the air contamination will be prevented.
Pastors discovered that the disease pebrine in silkworm was caused by a protozoan parasite, mainly at 1865 and all. And this infection, silkworm infection, was created a problematic issue in the silkworm industries. During this time, the pastor discovered that this infection is caused due to a protozoan parasite. And he introduced the use of complex media. This complex media is also called as culture media for the cultivation of bacteria. His accidental discovery showed that chicken cholera bacteria left out and bench for weeks lost their pathogenicity but could still protect bird from future infection. This led to the development of attenuation and live attenuated vaccine in 1818. The pathogenicity means the disease causing capacity of bacteria is called as the pathogenicity. And also Pasteur prepared an anthrax vaccine by heating attenuated anthrax bacilli in 1881 and coined the term vaccine for such prophylactic preparation. So the name vaccine was coined by this Louis Pasteur, and he also developed rabies vaccine in 1888. And this is the picturization showing that the boy is taking rabies vaccine.
And the another contribution of the Louis Pasteur is he disproved the spontaneous generation theory. Earlier in Greek philosopher Aristotle proposed theory of spontaneous generation. According to Aristotle, he is telling that life can arise from non-living matter. But Louis Pasteur, by his swam neck bottle, he disproved this spontaneous generation theory. And this is the picturization of Pasteur's experiment, how he disproved the spontaneous generation theory. In this picture, you can see a S-shaped or swan-necked flask. And this S-shaped part is the lid of the flask, and this lid can be removed when there is a need. In the Pasteur experiment, he took two flasks or lid containing broth. Broth are liquid extract where bacteria or microorganisms can grow well. In his experiment, first Pasteur sterilized the two bottles by heating. Later, he opened one of the flask lid and another one is closed. And after keeping the broth flask in open air for several hours, he proved that growth occurs in opened flask because closed jar prevent entry of air into the flask. In opened flask, when air enters the flask, microbes present in the airs will also enter into the flask. And by this experiment, Pasteur disproved the theory of spontaneous generation, concluding that life is not arising from non-living matter. Joseph Lister. Joseph Lister is known as father of antiseptic surgery. He was interested in prevention of post-operative sepsis. During his time, the post-operative sepsis was a major issue in the hospitalized patients because after the surgery, even though after the surgery is successful, due to this post-operative wound infection or sepsis, many people will lose their life. So he believed that the microorganism in the atmosphere might be responsible for post-operative wound infection because we know that now we know that the organism is present all over the environment. If they get any chance, definitely they will cause infection. And also mainly in the post-operative, mainly after the surgery and all, people will have wound in the particular site and also their immunity will be low. So automatically, easily they will get the infection. So in 1867, Joseph Lister introduced antiseptic technique in surgery by using this carbolic acid spray. And this surgical practice marked a significant milestone in the field of microbiology. So this introduction of this carbolic acid spray during operation and on wound after the post-operative surgical procedure has saved so many lives during that period. So this Joseph Lister is considered as the father of antiseptic surgery by this invention of this carbolic acid spray in the surgical ward and also after the post-operative procedure, mainly in the wounds. Robert Koch. Robert Koch is known as father of bacteriology. He was first to isolate Anthrax bacilli in pure culture and show spores in Anthrax bacilli. Anthrax bacillus means Bacillus anthrax. There is a spore-related bacteria. He introduced staining technique by the use of aniline dyes and also he implemented method of obtaining bacteria in pure culture by using solid media. So he only introduced solid media in the field of microbiology and obtaining bacteria in pure culture means when there is a pure culture, that means the single type of organism can be seen in pure culture of the bacteria. And he discovered tubercle bacilli. Tubercle bacillus means Mycobacterium tuberculosis and cholera vibrio, that is Vibrio cholerae. And also he discovered tuberculin. Tuberculin is an preparation from the tubercle bacilli that is earlier used for the skin test for tuberculosis. Coach postulates. Robert Koch postulates is having a very important significant role in the golden age of microbiology. According to Koch, the microorganism should be constantly associated with the lesion of the disease. It should be possible to isolate the organism in pure culture from the lesion of the disease. Inoculation of such pure culture in suitable laboratory animal should produce a similar disease in animal and it should be possible to re-isolate the organism in pure culture from lesion produced in experimental animal. Here in the picture, we can see this is an infected or died animal. In simple, according to Coast postulate, in simple, we can say that this infected animal sample is collected, and with the help of culture media and microscopy, it is possible to identify the causative agent responsible for the infection. In second step, what we will do, this cultured organism is injected to an experimental animal, then that experimental animal will reproduce the infection of the infected first animal. This is the first animal. That means both the infection will be seen. And if again we are collecting the sample from this infected experimental animal, we can re-isolate the organism in pure culture. That means these Coast postulates have proved to be and useful in confirming and doubtful claims made regarding the causative agent of infectious disease. Paul Ehrlich. Paul Ehrlich is a Nobel Prize winning German physician and scientist who worked in the field of hematology, immunology, and antimicrobial chemotherapy. He is called as the father of chemotherapy due to his various inventions, such as he first launched the search of magic bullet. In 1919, he cured trypanosomiasis in rat and mice by using tryptophan, and red and etroxyl. In 1906, he discovered salvarsan, destroying the spirochetes of syphilis. He introduced the method of standardizing toxin and antitoxin. He proposed the side chain theory of antibody production. He was the first to report the acid-fast nature of tubercle bacilli, and he developed techniques to stain the tissue and blood cell. And these are the contribution of Paul Ehrlich. Due to this contribution, he is considered or called as the father of chemotherapy. And in this chapter, we discussed only very key points and also the contribution of some important scientists such as Louis Pasteur, Robert Koch, Joseph Lister, and Paul Ehrlich. History of microbiology is an interesting, and we can say that it's an endless chapter with so many contributions. And in this chapter, we covered important points in the golden age of microbiology. Okay, also we can say that we focused mainly the golden age of microbiology, and these are some achievements in the history to remember.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.